Ladies and gentlemen, as it is time, we would like to begin Professor Kazutoshi Mori's lecture. We would like to introduce Professor Hidero Yoshida, Professor of University of Hyogo, who will act as the moderator of this session. Professor Yoshida, please.
So let's start a very exciting session now. So time is very limited, so I briefly introduced Dr. Kazutoshi Mori. He was born in 1958, Kurashiki, Okayama Prefecture, Japan. He was graduated from the Kyoto University, Graduate School of Pharmaceuticals. And then he became an assistant professor in Gifu Pharmaceutical University, but he decided to discard the permanent job to join the laboratory of the Dr. Joe Sandbrook and Dr. Mary Jane Gessing in the UT Southwestern Medical Center. And then he started the analysis of the unfolded protein response, which is a very fundamental issue in the cell biology. After 4.5 year research, he went back to Japan to join the HSP Research Institute as a principal investigator and continue the research of the unfolded protein response. Then he became an associate professor in Kyoto University, and finally, now he is a full professor in Kyoto University, Graduate School of Science. He was awarded with various prestigious awards, scientific awards, including the Wiley Prize, Gardner Awards, and the Shaw Prize, and the Lasker Award 2014. He was awarded with Medal of Honor with Purple Ribbon from the Emperor. So, title of his talk is, how did the Lastic Country Boy become a Lasker laureate? The talk will be very, very interesting. So, Dr. Kazutoshi Mori, please.
Thank you, Dr. Yoshida. It's my great pleasure to be here. So let's start my talk. My title of my talk is How Did Lastic Country Boy Become a Lasker Laureate? Keyword would be ambition, preparation, challenge, patience, and health. As Dr. Yoshida mentioned, we are here in Tokyo. I was born in a small town facing the Seto Inland Sea. Okay. So now, in 1958, so this main island and Shikoku Islands are connected by this beautiful bridge in 1988. My family has no background of, no atmosphere of science, but at elementary school days, I loved to read comics, like Mighty Atoms, Astro Boy in American movie, and Maha Go Go Go, Speed Racer in American movie. I also like to read mysteries, like Sherlock Holmes and Lupin. So, who solves a mystery in science means a researcher. So my first ambition is to become a scientist, researcher, a PhD scientist. I also like to read a newspaper from elementary school days. When I was a junior high school student, newspaper told me that the discovery of quarks. So I knew atom is composed of nucleus and electron, and the nucleus composed of proton and neutron. But I was amazed, but further smaller things exist, like up and bottom and strange. So I was curious how small things can be, so and how many quarks would be there. Later, I learned that Dr. Kobayashi and Masukawa predicted the total 6 quarks. Then, later, charm and bottom and top quarks are discovered, and as their prediction was right, they got a Nobel Prize 2008. Unfortunately, Dr. Kobayashi is not here, but he's very busy. So then, subsequently, I learned that first Nobel laureate, Dr. Yukawa, and second Japanese Nobel laureate, Dr. Tomonaga, both are graduates from the Faculty of Science, Kyoto University. So my second ambition was I want to go to Kyoto University to learn physics. At that time, even at high school days, I did not like biology at all, because textbook says that this is animal cells, this is plant cells. Some common things exist, like orange nucleus, blood Golgi apparatus, and blue mitochondria. But plant cell has something else. Green chloroplast, blue vacuole, and plant cell has cell wall. And so bacteria, E. coli bacteria does not have this kind of things. So everything is descriptive. I thought that I had to remember everything by my heart. So I was much more interested in the physics. So I studied hard to enter university, Kyoto University. Then, when I was last year of high school, I found that I'm in the border to pass the examination. I may pass examination, but I may fail to pass examination. So at that time, I was a really local boy. No private school was there, so I decided to not take a chance. I chose an easier way. So I entered Department of Chemistry, Faculty of Engineering. So this entrance examination was a little bit easier than entrance examination to the Faculty of Science at that time. After oil shock, chemistry was not attractive to many people. So then my choice was right. I could enter directly to the Kyoto University. But kind of I had a disappointment. I gave up my dream to become a physicist. But then still I would like to read a newspaper. Then, to me, this newspaper suddenly started to describe the work done by Dr. Tonegawa. I was a freshman of Kyoto University in 1977. Then, 10 years later, Dr. Tonegawa got Nobel Prize in the first Japanese Nobel laureate in the physiology of medicine field. Then, because from description newspaper, I met molecular biology. I had many happy meetings that changed the direction of my career. Particularly 3 meetings is important. First one was a meeting with molecular biology. Do you know DNA is a genetic material, the molecule that carries heritable information. Molecular biology says DNA consists of 4 letters, G, A, T, C, contains information how amino acids are connected to make proteins. Then, so for example, ATG means methionine, amino acid methionine. Surprisingly, this genetic code is conserved in all species on Earth. ATG means methionine in animal cells, plant cells, or in bacteria. So I was amazed by the fact that biology has a basic and simple rule. So I did not like biology, but molecular biology is very interesting. And because this genetic code is conserved in all species, so if you take human gene from human cells, they put human gene into bacteria. Because genetic code is conserved, this bacteria produce human proteins. And this bacteria divides every 20 minutes. That means we can have a mass production of human proteins in E. coli. So, to me, molecular biology is very interesting and useful. They can harness people. So, I wanted to do molecular biology. So, I want to be closer to biology. So, fortunately, I could move to the Faculty of Pharmaceutical Sciences. I could switch from chemistry to biology. But molecular biology are very new techniques. Only few laboratories can do molecular biology in Japan at that time. So no molecular biologists exist in this Faculty of Pharmaceutical Sciences. So I started my career from biochemistry. But research itself is very interesting. Until entering the university, we are solving a question which has only one answer. But in the research, we try to solve a question which has no answer. Nobody has an answer. So that research itself is interesting. So I studied hard. I had a good preparation days and obtained PhD in 1985. So I want to stay in academia. But the problem was that no postdoc system in Japan at that time. Many PhD scientists, if PhD scientists want to stay on academia, many of them stay in the laboratory by doing part-time job. It's called over-doctor problem at that time. So I did not want to do over-doctor. So because this is pharmaceutical sciences, my seniors in my laboratory almost all go to pharmaceutical company. So I was wondering how to do, but very fortunately, associate professor in my laboratory became a full professor at a local university called Gifu Pharmaceutical University. Then he could get one position available, assistant professor job is available. So he recruited me to his laboratory. So I could I had a permanent position at the local university. So this was very fortunate. So I worked hard, but still, here is a biochemistry only, no molecular biology. But I worked hard, and I published 8 papers in 4 years. This is good preparation to write the papers. So, but I worked hard for 3 years, but I got result which published in the cancer research. That's quite a good journal, but I thought that this is the end of the research. No more good future for this project. So I told my boss, let's stop this project, do something else. But boss said, no, this is interesting. You should carry on. So he's a boss and my assistant, so there are no way to reconcile between us. So I want to do something more interesting and something more important. So I decided to quit this permanent job to go to United States to learn molecular biology. I sent 3 letters to people in East Coast or West Coast, but they did not accept me. So only Joe Sambrook and Mary Jane Gasing at Dallas, Texas accepted me as a postdoc. Joe Sambrook is a famous scientist, famous as one of the authors of the book called Molecular Cloning. At that time, this book was called Bible for molecular biology. So if I go to this laboratory, at least I can learn molecular biology and English. Then decide to go to somewhere else. But second happy meeting happened here. I became postdoc in their laboratory in April of 1989. Then I encountered the unfold protein response there. That changed my entire life. Since then, I was working on this response for nearly 26 years. I was like a postdoc like this. Then, after last year, finally, I got Lasker Award. This lady is the president of Lasker Foundation. Do you know this gentleman? This Dr. Joe Gostin, chairman of Lasker Award jury. I think he's the greatest medical scientist in the world now. He's my hero. Suddenly, here Japanese, but June 24th, I got an email from his assistant. Joe Gosling asked me to send you the attached letter. I opened the letter. Here. Dear Kazutoshi, as Chairman Lasker Award Jury Committee, it's a great pleasure to inform you that you, together with Peter Walter, have been selected to be the recipient of 2014 Lasker Basic Medical Research Award. What a big news to my heroes. Her discoveries concern unfold-potent response, an intracellular quality control system that detects harmful misfolded protein in the endoplasmic reticulum and then signals to the nucleus to carry out corrective measures. Does this make sense to you? Only makes sense to specialists, so I explained what is this. There are three backgrounds. First background for my work is molecular biology part. A DNA contain information how amino acids are connected to make protein. That means protein is a linear polypeptide. Amino acids are connected in a linearly. It is just like a string when newly synthesized. But we are living in a three-dimensional world. String cannot perform its job. Each protein has its own three-dimensional structure. It must gain their proper structure to achieve their function assigned by genetic code. For example, insulin is a linear polypeptide, but it must have this kind of structure. You know paper folding to make a crane. So this is a paper folding, but this issue is called protein folding. If insulin is properly folded, it can bind to its receptor. Then it can decrease blood glucose level, so you can be healthy even after a meal. Blood glucose level is decreased. But insulin, if insulin is not properly folded, it cannot bind to receptor. That means blood glucose level remain high. If such situation continues, such person will develop diabetes. So protein folding is quite important for ourselves. And the ribosome is a place where protein is synthesized. But there are 20 amino acids, but some amino acids are hydrophobic. That means very sticky. The sticky amino acids stick together to make misfolding. This is a misfolding, not properly folding, or aggregated. So inside cell, protein folding is not easy because of the high concentration of proteins. To solve this problem, we have special type of proteins called molecular chaperones that bind this sticky amino acid to help proper folding. This is folded proteins. Okay? Chaperone is not a special word, probably new to most of the Asian country people, but chaperone, she is a chaperone. Person in the past who went with a young unmarried woman to social events like a dance party. This is a lunar painting, but to make sure that woman behaved properly. Then if she is maturated, she get away. Molecular chaperone is doing the same things. Protein easily can be misfolded, aggregated, behave improperly. So to molecular chaperone assist protein to behave properly to get folded. This is the function of chaperones. Second background two for my work is the cell biology part. As Dr. Yoshida yesterday did a flash talk and did some introduction, if you look at cell by microscopy, it's very simple. There's a nucleus containing DNA, the Golgi apparatus, and mitochondria produce energies. But if you look at cell by electron microscope, that landscape was dramatically changed. cell contains many organelles, membrane-bound space called organelle. Peroxisome, ribosome, then largest one is the ER. If we use GFP, like ER looks like this. Here is a nucleus. The ER is a mesh-like structure spread all everywhere in the side of the cell. These prominent scientists at Rockefeller used micron electroscope to search research in cell. Then Dr. Claude and Dr. Dedieu found pyroxome, lysosome, and Dr. Parade found ribosomes, a dense structure that makes protein. And then insulin, hormone like insulin, is synthesized inside the cell, but is in the bloodstream. How they are synthesized and then go to the nucleus outside. They found that, of course, insulin is a protein synthesized in the ribosome, but not synthesized on the free ribosomes. It's a ribosome bound to the ER membrane, then go out to the cell through Golgi apparatus. Because of these distinguished discoveries, they got Nobel Prize in 1974. I think this is the first generation protein research after 50s. Then next question is why insulin is synthesized on the membrane-bound ribosomes, not on the free ribosomes. Dr. Grobel discovered that insulin hormone has a signal that hooked up with this ribosome to the ER membrane called the signal theories. Then his prediction was correct. He got Nobel Prize in 1999. This is the third generation research from 70s. The researcher from 80s did how protein moved from ER to Golgi and Golgi outside. They found protein is packed in a small vesicle, then moved from ER to Golgi, then so-called vesicular traffic. 3 eminent professors got Nobel Prize 2 years ago. We think these science are third generation. Now we are on the fourth generation, working on the protein folding issues. So, last background for my work, ER part. Although Hidero is already mentioned, ER is a protein producing factory. They have ribosome attached to the ER membrane, and this ribosome is a kind of belt conveyor. Synthesize a protein, deliver protein inside the ER. ER contains many molecular chaperones that assist productive folding. And folded protein can go to final destination through a vesicle. Then usually, more than 90% protein gets properly folded and go to their final destination. But because cell synthesize many proteins, some proteins stochastically misfolded, unfolded. But this unfolded is quite problematic. So somehow this unfolded protein are recognized and get delivered back to the cytosol to get degradation. So ER checks the quality of protein. It's folded. If folded, it's okay to go. But if it's not folded, unfolded protein are degraded. But this protein quality control system can be compromised under a variety of physiological or pathological conditions. So such time, there are many more unfolded protein in the ER. This is problematic, very critical, because only folded protein can go to the final destination. This situation continues, we lose functional protein at the various destinations. But very fortunately, we have a good system to encounter this. This condition is called ER stress. ER is stressed. We have a system to encounter ER stress. That's the unfolded protein response I've been working for 26 years. To cope with this stressful conditions, cells induce increased level of molecular chaperones to facilitate more folding, or induce these destroyers to enhance the capacity of degradation. So now cells equipped with homeostatic responses. This is unfolded protein response. This is what my boss discovered. Now I'm coming to this field. Story again. Usually, ER has a protein control system for folding or degradation. But if we have more unfolded protein in the ear, cell induce increased level of chaperones or induce destroyers to facilitate folding or facilitate degradation. This is what discovered my boss discovered. But no mechanism at all at that time, in 1989. So we looked for the mechanism. What is necessary for this happen under ER's condition? You know what is necessary? Of course, here is I is necessary. Somehow, ER must sense that this stressful situation. So we need a sensor in the ear. To search this sensor, I use E cells. This is both direction. They used to work on the mammalian cell, but many people working on this system, but people did not get good result because mammalian cell system is much more complicated. So both decided to go to more simpler system, to go to E cells. Their decision was right. Then I did engineering. Usually E cell is like a white. But if this unfolded protein response is activated, I make this E cell turn blue. But if this unfolded protein response is not functional, this E cell remain white. It's kind of simple blue or white selections. So I mutate DNA randomly using mutagen, then screened 100,000 colonies. Most of them are turned blue, but I found only 3 colonies remained white. Then pull out a gene defective in this clone. In 1993, I published a paper which discovered a sensor molecule called IR11. Then, what else is necessary? Only molecular biologists understand, but these are induced, but under normal condition, these inducible factor is not walking around. It's kind of sleeping in the nucleus. So in the case of ER stress, somebody hit a drum, wake up chaperones, go to ER to help. So we discovered this drummer in 1996. And also, there's many drums here. So I discovered first thing was, drum is here. If we hit this drum, chaperones wake up. Then, of course, ER and nucleus is a different compartment. This ER stress signal must be transmitted to the nucleus. We saw the mystery in 1997. So I found this drum in 1992, but then find the sensor, but almost at the same time, Peter Water came in. He found the same sensor molecule almost at the same time. He is, his boss is Dr. Blobel, second generation scientist in protein research. He got Nobel Prize in 1999. So his PhD work pushed very strongly to Dr. Blobel to Nobel Prize. So he is already a famous scientist. He may look old, but he is not so old. He is older than me only by 4 years. So when I did this work, I was mid-30, and he was late 30, but he was already famous professor. He already been a professor nearly 10 years. top runner of cell biology research. He was interested in this system, jumped into this field. So we found a drummer in 96, and he also found same drummer in 96. So during 90s, we had a very severe competition with a big guy, Peter Walter. So next 5 slides are only for molecular biology to understand. I need to balance all both kind of explanation and more detail. So IRE1 is a sensor molecule, transmembrane protein in the ER. Then somehow, when unfold proteins accumulate, this oligomerize, then trans-orthophosphorylate to be activated. Then DRAM is a sixth acting element in the promoter region of chaperones. DRAMA is a transcription factor. How this IRE1 and HAC1Ps are connected is there is a very unusual mechanism. IRE1 mediated splicing of HAC1 mRNA encoding this transcription factor. Both Peter and I discovered HAC1 and the splicing almost at the same time. Okay. This is a Western blot of HAC1 protein. Under normal condition, no HAC1 protein is detected. But after years, we can clearly detect this HAC1 protein. So HAC1 protein regulation is a level of amounts. Okay? If we check its mRNA for northern blot, there exists, even under normal condition, HAC1 mRNA exists, but no protein is detected. But after years, this mRNA becomes shorter. That means gets spliced. Then we sequence what's the difference here. After years, these 252 nucleotides are removed in IRE1 dependent manner. Then because this 5' region is located in the ORF, usually precursor mRNA encodes 220 part plus 10 amino acids, total 230 amino acids. But after removal of this intron, this 10 A region is eliminated. This 220 amino acid region is connected to 18 amino acid region to make 238 amino acids. Both Peter and I see completely same phenotype, but our interpretation is completely different. You understand? There are the two interpretations. Issue is this precursor mRNA is translated or not translated. Peter says this mRNA is translated, but not protein is detected. That means this protein is synthesized, but it's rapidly degraded. That's why we don't see protein under normal condition. But after years, this 10 amino acid, destabilizing 10 amino acids removed, but this 18 amino acids not have such effect. So this protein is stable, and we can detect this protein. That's Peter's story. But my story is this messenger RNA is not translated. That's why we don't see a protein. But this protein is translated. That's why we see this protein. So we had a big argument, but of course, Peter's a famous big scientist. I'm a kind of still postdocs. Nobody believes our story. But only I and this young guy working with me believed our story. We worked hard for more than half years with less sleeps. And then finally, we never gave up. Turned out that our story is right. This mRNA, precursor mRNA is not translated because this intron has a bulky structure that blocks translation with mRNA. Peter also realized that he made a mistake in his interpretation. He corrected his story by himself. So that's my hard time. During 90s, we had a severe competition, but we never give up. Kept going. One thing we got, one more thing I had good suggestion from boss is after Hidero said staying 4.5 years in Texas, I came back to Kyoto to find a postdoc position again at the HSP Research Institute. Dr. Ira was the head of this institute. I had IRE1 gene. So next target was this transcription factor. So usually, now, usually somehow this IRE1 signals transfer to the nucleus. But if we don't have IRE1, no signaling occurs. The most usually is people do multi-copy suppressor screening to get next gene. It's a quite easy experiment. So just put this gene in the multi-copy level that suppress this no signaling phenotype. So I said, when I joined his laboratory, I told Dr. Ura, I'm going to do this multi-copy suppressor screen. But Dr. Ura said, he's a gentleman, but said, no, you shouldn't do that. Because if you use this suppressor screening, there may be many, many genes here. You may end up obtaining many, many genes between IRE and this putative transcription factor. You will never be able to get this transcription factor. So you should think of a method by which you can get this factor directly. It was a very hard order. So I struggled for 1.5 years. But finally, I came up with a good idea of one-hybrid screening. This is called two very special techniques. So if you want more detail, I'm happy to explain to you after this lecture, not in this time, because most people fall into sleep during my explanation. So then this is my third happy meeting is like with one-hybrid screen. Peter used this multi-copy suppressor screen because he's a normal yeast researcher. But if you use this system, this works only for yeast gene. But one-hybrid screen I developed is work for any species. So Peter is a great scientist. He published many, many great papers in the top journal, but he stayed on the yeast system. But because of this development, we could move to the mammalian cells. And I had a talented postdoc colleagues, Dr. Yoshida. He was in the same institute. And he recruited to my project. But he is brilliant. And he found a drum in 1998. And using one hybrid screening system, he got a sensor and he got a drummer in mammalian system, which Peter could not do at all. So now close to the end. Now, yeast has only one sensor, but during evolution, number of sensors increased. This non-vertebrate, C. elegans, Drosophila melanogaster, this kind of non-vertebrate has 3 sensors. But vertebrate, like fish and mice and we human, has total 5 sensors. So this UPR system is very important to maintain the homeostasis of ER, because ER is a place for maturation of secondary and transmembrane protein, like hormone and hormone receptor. It's very important for cell-cell communication. So this system must be very, very functionally well to maintain our bodies. Now, we have 3 sensors called PARC, AT6, and iron, we discovered. The physiological importance of UPR was discovered by making and analyzing knockout mice. David Long at New York at that time found that if mice do not have this PARC gene, they can be born, but they develop diabetes. Because pancreatic beta cells is a factory to produce insulin. So to maintain the function of pancreatic beta cell, this PARC-mediated UPR is very important. If you don't have IRE1, this is consult from yeast cells, mouse cannot be born. They died during development. Rory Graham at Harvard discovered that liver could not be developed well. because liver is a producing factory for many protein in the blood, circulating in the blood. So this system is also important. That we discovered 86, the third sensor molecule, only operating in a vertebrate cell, not in a non-vertebrate cells. So this has two genes. Single knockout has no problem, but we have a double deletion called embryonic lethality. Mice die very early days. So because mice die very early days, we could not figure out why mice dies. So we switched system to fish system called Medaka fish system, Japanese fish system, to find that 86 function is important to develop on the notochord. It's a cartridge-like a structure before the bone is formed, backbone is formed. So all of these UPR systems important for our development. In addition to the physiological importance of early development stages, this UPR is reported to be associated with many diseases. Once our basic researchers found a mechanism operating in the UPR, many medical doctors get interested, see what's going on, his disease. Then not only diabetes, like Parkinson's disease, atherosclerosis, inflammatory bowel disease, many diseases may be developed by the problem ER. So that's why I got Lasker Medical Award. So this is the last slide. I talked my career. So I'm now successful, but my keywords were ambition, preparation, and challenge. Patience. The most important thing is probably health. You need to be healthy to achieve something, but to do work hard. Both mental and physical health is quite important. Yamanaka-san is a judo player, and I'm a kendo player. So using bamboo sticks and protectors. I've been doing kendo since my junior high school days. That helped me a lot. Especially, my life is like a tightrope walking since I went to the United States. But I need to always go forward to maintain motivation high. This kendo helps me a lot. But you have your own goals. But not so many people can get to your goal directly. There's many barriers, and then, as I told you, I had a very winding road. But okay, DNA is a helix. So if you keep look up, you may get to your goal someday in your near future. Thank you very much for your attention.
Thank you, Professor Kazutoshi MORI. So his talk contains a lot of suggestions for young investigators to survive in a very hard situations. So you may have a lot of questions. So it's a time for discussion. Oh, yes.
Thank you very much for this nice presentation. My name is Yasser Gaber from Egypt. I have 3 questions. First,
one by one, not
okay. I want to know, does any of these receptors have been structurally determined? Does any of these systems, UPR, is structurally determined?
No. Yeah. Whenever I made a seminar, many question comes, how these sensor molecules detect ER stress unfold protein. It's no answer yet, because crystal structure is very difficult to make, because ER protein are very sticky compared with protein in a cytosol. Only 2 success here Okay? There are many, many sensors there, but only two success only for this luminal region of east IR even. And luminal region of human IR even is solved, but not consistent. Okay? So that's a problem. Maybe a packing structure problem, or maybe sensing system may be different. But when Peter water get crystal of the east IR1, this get dimerized, there is enough pockets for amphor protein to bind. That's his story. But Randy Kaufman get crystal from human IR1, but he doesn't see enough space for binding amphor proteins. So no, there's no, because of no crystal information. So we don't have enough information for how the sensor gets recognized ER stress. Okay?
Okay.
Okay.
So this may answers my second question. Is there enough information about how unfolded protein interact with these receptors?
Yeah, that's very difficult things. Because still, because to search molecules involved in UPR, people use drugs, chemicals that artificially make protein unfolded, misfolded. So like a tunicamycin, some chemical that artificially malfold many, many proteins at the same time. But in the situation that happened during the novel development, not such a heavily destroyed does not occur in the development notocode. This notocode synthesizes large amount of extracellular matrix protein, like a collagen, you know, collagen. Okay, so this making particular type of protein is physiological inducer of ER stress. So now we are starting to understand what, now I'm going to start to understand what kind of molecule cause physiological ER stress. Then we need to work on why this is sensed by this 8006. But no, it's a next future question.
Okay, so the last one, is there a kind of hope that this discovery can help in treatment of Alzheimer by activation of
yeah.
mechanism?
So Alzheimer is a huge problem in all over the world. And some people say ER stress associated development of Alzheimer's disease, but still there's an argument. It may not be a cause. It's a result of Alzheimer's disease. Development of Alzheimer's disease may cause ER stress. So there is argument. So I try to, because of in Japan, or as Prime Minister Shimomura says, Abenomics, economical growth is very, very stimulated. So I'm thinking about the UPR to harness people, but still we need to do a lot of things because these kind of things are cell type specific, phenotype is cell type specific. Okay? These sensors are ubiquitously expressed. All cells have all three sensors, but pancreas beta cell rely on IR PARC system. This PARC beta cell has 86 IR1, but still suffer from diabetes. But in the case of liver, this liver has 86 and PARC, but still suffer from ER stress because lack of IR1. Then notocode suffer from ER stress in the absence of 86, but in the presence of IR1 and PARC. We don't know why this kind of difference occurs. We need to do much more basic research to harness people. So now we are trying to make Parkinson's disease model using this kind of double knockout mice. Then if Parkinson's disease happens, we may be able to think about curing these mice. So also in the cancer cells grow in very stressful conditions. If they grow inside a body, they lack oxygen. or they lack nutrient. That causes ER stress. So they utilize this UPR system to survive. So if these gene systems are knocked out, cancer cells cannot grow in a nude mice. So now many people are looking for the chemical that block cancer progression by inhibiting UPR. So, but some company developed nice chemicals to block this PARC system. But it kills cancer, but its side effects severe because pancreatic beta cell needs PARC activity. So PARC, this severe side effect on pancreas, that's a problem. So now we are doing some this screening to block this IR1 system to hopefully to kill cancer or virus infected cell. Virus also use ER to their propagation. Okay.
Any other questions? Oh, yes, please.
Thank you very much for your excellent talk. I'm Lee Han-jang from University of Tokyo, and I want to know more about, in terms of how to compete with other laboratory, especially how to compete with big laboratory. And in your lecture, you mentioned that you have a mammalian system, which the Professor Peter Walter's group couldn't. But I also think after publishing, and they can duplicate your result ideally, and eventually big laboratory takes all. So sometimes we feel that, like in the United States, they are really powerful, and so yes, I want to ask that.
Because it's a good question, and then because this UPR system is intracellular signaling system. Because previously, signaling come from the surface of the cell. The receptor gets some ligand, then sends signal. These are well studied, and so if we work on this system, probably people in the United States can work much more faster. But this is intracellular communication between E and nucleus. This system is completely different, used as a kind of new way to communicate. Like splicing is very special event. It's not utilized from signaling from cell surface to the nucleus. Only occurs between E and nucleuses. Then, and also, we discovered this IRE1 system. IRE1 system, when signals occurs, this mRNA becomes shorter. So it's obvious. We can see difference in the size easily by northern blot. But this system is conserved in mammalian system. But IRE1 is conserved. It's very similar protein. It's very similar human protein exists in a human mammalian cells. But this HAC1 does not exist in mammalian cell. There are no, even a multicellular system, like after C. elegans. This HAC1 exists only in yeast cells. Then people search for this mammalian version of HAC1, but nobody identified this HAC1 because here is an intron 252 base pairs, but in the mammalian cells, intron only 26 base pairs. You cannot see difference in the size by northern blot. Hidero was very talented. Somehow he put out. It took maybe 5 years, but nobody understand what. Nobody could identify this mammalian version because secret was size of intron was too small to see. But finally, he using one hard system. He got 2 transcription factor. One is XBP1, one is 86. Both are very interesting molecule, but very, very lucky to us. Its regulation is very unusual. So XBP1, case of XBP1, mRNA is regulated, but difference only 26 there. Very special eyes is necessary to find out the secret. Also, 806 is very interesting molecule, and then this 806 found to be a transcription factor, but it takes some time to figure out what's going on, but it synthesizes a transmembrane protein, synthesizes as a sensor. Then, but this cytosolic region has function as a transcription factor. So if ESS occurs, this protein gets cleaved. Then cytosolic region is liberated from the ER. Then go to the nucleus to activate transcription. So because this is a kind of new field, the new mechanism is utilized. We could keep competing. So if we work on the normal, usual signal cascade, but we cannot keep up with Peter Walter.
Any other questions? Yes, please. And then next.
Yeah, I have a question. Did the unfolded protein response sensors have selectivities for the specific protein? Because if a protein have problem with sensors, all the proteins can now be correctly folded.
Special protein.
For the selectivity for special protein. Or
Yeah, so that's your question connected with your question, right? Third question. So now we started thinking of answering your questions. Why certain protein is promoted to the certain cells? So we don't have enough answer. Why collagen is why mass production of collagen activate 806? Then in the case of notochord, they need notochord to develop. They need extracellular matrix protein to support cells, to align smoothly. But if they cannot make collagen, alignment was disturbed and they stop growing. Then, but question is why 806 is activated by the mass production of collagen has no answer yet. We try to answer a question because still we don't have activation. Still we do not know enough for the mechanism of activation of 806. Then we don't have a crystal structure. So that's our problem. Takes more time.
Thank you for giving us interesting presentation. I'm Takaki from the University of Tokyo. And my question is, why were you able to quit your permanent job when you are young?
Because I was young. I could be desperate. If I have old enough, I have kids, I have no way to get desperate.
If I were you, I want stable life, so it's a very difficult decision to go abroad. Did you have any confidence to successful in the U.S.?
No, that's just a problem, personality problem. Just choices, you want a permanent. So you cannot have many choice at the same time. You want a stable job, you want exciting results. But so just I think I suggest young people that make a ranking of your wish. Which is first wish? First wish is stable job. First wish is exciting result. So if you wish stable job, you can stay. No problem. I don't blame you. I want to have an exciting result because experience is difficult. Everybody experience. You have to work hard to get master degree or PhD degree. Anyway, you have to work hard. If you need to work hard, I want to get interesting results, exciting results, important results. That's a motivation. And also, That time, USA is like a kind of a fascinating world. More than 25 years ago. So someday I wanted to go to United States because many professors, associate professor, assistant professor go to United States with keeping his job if they are successful. But someday you have to go to United States. But at that time, if so, I want to go to United States at young days. And also Dr. Tonegawa. Dr. Tonegawa affected me many things. He said 30 days is very important. You are 30, from 30 to 40, it's very imaginative days. So you should, he said, you should not follow your order of your boss. You should try your talent. So I took a chance.
Thank you very much.
I'm here.
So that is a secret of his success. So I'm sorry, you can discuss later. So time is up. Thank you again, Dr. Kazutoshi Mori.
Thank you very much, Professor Mori and Professor Yoshida. Amusing lecture. Now, this will be the end of our morning